Hi kids, so now we proceed to the second part of our discussion in your topic 6 and that is all about chromatography. So nalalantay duha na nabilin, chromatography and your mass spectrometry. So what is chromatography? Chromatography is an analytical technique commonly used for separating a mixture of chemical substances into its individual components so that the individual components can now be thoroughly analyzed. So basically, chromatography is all about separation. Mo man na siya nga to ang ginatanaw. Now, you might wonder na ma'am, the word itself depicts color, 'di ba? Kaya chrome or chroma means color. Because if you try to go back to history, the very first thing that kung kinsa man to naka-discover, I forgot the name, basta katong pinaka-first na buhat aning chromatography, ang separation man gud na iyang ginabuhat is with the use of pigments. And syempre, pigments have colors, thus na-coin in the word chromatography. So, what are the basic components of chromatography? So number one, you need to have a mobile phase. Mobile phase is also known as your carrier. Now, a mobile phase could be a gas or liquid, and this is where your solvent moves through the column. And other than that, mobile phase kaya siya tinatawag din na carrier because it is actually responsible in carrying your sample. Next is your stationary phase or also known as your adsorbent. Stationary phase can be solid or liquid, and it is where your mobile phase flows. Now, from the name itself, stationary phase, stationary, meaning it does not move, it is fixed, and it stay fixed, kumbaga, inside your column. So, what is this column now? Column holds your stationary phase. So, kumbaga, to give you a picture, namoy column, that is where you place your stationary phase. Stationary phase remain fixed or does not move, kaya stationary, naalang yun na siya dara sa inyong column. Mobile phase na will enter inside your column. And kaya dyan nilalagay yung sample because of course your sample needs to move all throughout your column so that separation may occur. Next is your eluate. Eluate is what we call your separated components. So once na separate ng mga components again, eluate ang term, then you have your eluent. So careful ha, eluate, eluent. Eluent is the fluid or substance that enters the column and now moves your analyte. Now, syempre, si mobile phase carrier lang, what would actually help in the movement of your analyte and in the separation is your eluent. Now, other terms that you need to know and understand is your elution. Elution is the process of washing out a compound through a column using a suitable solvent. Then we have your analyte. So I know you are familiar with the word analyte. Analyte is the mixture whose individual components have to be separated and analyzed. And then lastly, your retention time or factor. So this means the time it takes for a compound or analyte to elute or the time it takes for your analyte to be separated. So to give you a general view as to how your chromatography works, let us look at this one. Kaning butanganan kids, kani in itself, that is your column. Kaning material inside, sa kaning naay material inside or kaning colored dots ninyo makita, these are actually beads. That is your stationary phase. Your mobile phase is where your that is what will pass through your column carrying your analyte. So kani makita ninyo na shaded diri na violet on top na line, that is your analyte. And then so ingani ang mahitabo. Once you place your analyte and then amoy makita actually diri kanina part kaning white na part here. This is actually your eluent na in order for your sample to move and then start being separated, you need to add an eluent. And then, after some time sa hinay-hinay from your original analyte, your components will now separate. And then, syempre, naman jud ay mauna na ay maulahi in terms of the separation, but in terms of who comes out first and who comes out last, that would depend actually on what are the substances that you fill in or you used, unsa ang nasa inyong stationary phase, unsa inyong mobile phase, and unsa to inyong eluent. And we will discuss that further later. So basta again, what I am trying to point out here is that for you to have in general na view lang yun kung unsa ning separation and chromatography is all about. So, mo ang imahitabo. So, let us take for example, inyuhang analyte is your blood. And di ba, blood has a lot of components inside. So, mo na in order to get one substance from the other, so muna na natay separation na buhaton sa ilaha. And unsaon siya pag-separate? So imagine, daghan taong pwede factors na isipon ani. It could be the size, di ba? Dako si glucose, gamay ang uban ninyo na mga substances inside the body, or it can be in terms of uh, electric charges, di ba? Proteins carries positive and negative charges. So mga ingana ang ato ang buhaton diri sa inyuhang chromatography. Now, chromatographic techniques may be classified according to their mobile phase. So, as mentioned, mobile phase can either be a gas or liquid, and thus naatay gas chromatography and liquid chromatography. So, let us start with your gas chromatography. Now, here in your gas chromatography, in terms of separating your compounds, what we are trying to look at here is actually based on their volatility. So when we say a certain substance is volatile, di ba, meaning it is easily evaporated at normal temperatures. Now, gas chromatography is useful for compounds that are naturally volatile or can be easily converted into its volatile form. Thus, dili tanan compounds pwede nato i-gamiton for gamitan og gas chromatography. Again, we need to consider these two, that they are naturally volatile or that they can be easily converted into its volatile form. Now, there are two types of gas chromatography. So you have your gas liquid chromatography and your gas solid chromatography. Now, gas liquid chromatography is actually based on partition, while your gas solid chromatography is based on adsorption. Now, ma'am, unsa ning partition? Unsa ning adsorption? These two techniques, these are actually separation techniques, will be further discussed later. So, ato lang po na siyang i-relate pag moabot na ta or mo-reach na ta ani na separation technique. Basta again, gas liquid is based on partition, gas solid is based on adsorption. So, this is how your gas chromatography would look like kung unsa ang iyahang mga different na components. So, mo na naamoy mobile phase or kani your carrier gas, and then dara ang inyuhang column, then naagihapon mo detector na amoy recorder. And for you to understand the overall setup of your gas chromatography, so let us watch this video. Okay, so that actually explains how gas chromatography works. So, 'di ba, kaya siya ginatawag na gas? Kay, as you can see in the video, ang nahitabo, your sample is being evaporated. And that evaporated na sample, natay actually collector dira na part sa inyuhang detector. The FID that you saw in the video stands for your flame ionization detector. So later ato nang i-discuss further. Basta mo na siya yung detector. So it is able to detect whatever sample was vaporized, and then it changes sa inyuhang any changes na mahitabo dira sa inyong FID that is being recorded, as you can see here na naamoy makita na peaks. So inyuha maning recorder, so it is being detected and then recorded, and then kanang peak na inyuhang makita kung unsa kataas ang peak sa inyuhang graph, that is already equivalent to referring already to the concentration of your sample. Okay, so syempre, do not worry, isa-isahon gyapon nato og discuss inyuhang basic components. So let's proceed. Now, one basic component of your gas chromatography is your column. Columns can either be packed or capillary. So natay ginatawag na packed columns, natay ginatawag na capillary column. If it is a packed column, it can either be a glass or a stainless steel. If it is a capillary na column, then thin fused silica usually ang ginagamit. Now, packed columns are filled with inert particles, such as your diatomaceous earth or your porous polymer or glass beads coated with a non-volatile liquid in your stationary phase. And the liquid stationary phase must be non-volatile at the temperatures used. Kaya ma-useless kung pati ang inyuhang stationary phase volatile po di ay kaayo. So it must be thermally stable and must not react chemically with the solutes to be separated. Kung mo-react na siya og apil, then pwede ta magka-miss kanang miss tawag na basta mamali ta og detect mamali ta og detect sa kung sa to na certain substance ang present. So again, this is the content of your pack column. So now let me discuss the difference between your pack column and your capillary column other than kung unsa siya made of. So na-mention man nato kaganina na si pack, glass or stainless steel, si capillary thin fused silica. So try to look at this image. Medyo literal ang naming ani sa inyuhang kanina types of column na when we say packed, it is packedly, grabe ka pack ang inside, it's either ang i-pack up dira sa hollow portion sa inyuhang column. It could be a bead column, it could be a porous layer, so katong mga na-mention kaganina sa inyuha. Whereas in your capillary, which is also known as an open column, since here, as you can see here sa hollow part sa inyuhang column, wala tayong gibutang na anything, what is done here in your capillary or open column is coating. So kaning makita ninyo na colors dire, kaning blue ug red kana, coating of the sides of your column. So mo na wall-coated open tube or porous layer open tube. Now, pack column can actually be used pag mag-gas liquid chromatography mo or mag-gas solid na chromatography. But for your open capillary, this can only be used if mag-gas liquid chromatography mo. Okay? Now, capillary could actually provide a higher separation efficiency compared to your pack na column. But the problem lang yun with capillary is that it tends to be overloaded easily. Na kumbaga, dili daghang sample kadayon na pwede i-measure. So, mao po na ang actually ang advantage ni pack. Si pack kaya siya greater sample capacity. So, next, so, dili na tumupasok si FID. There are actually two detectors that can be used in your gas chromatography. One, we have your thermal conductivity detector, and then you have your flame ionization detector or katuning FID. So, for thermal conductivity, it contains wires or filaments that change electrical resistance with change in temperature. Whereas, your FID is actually more sensitive than your TC. And ang ginagamit here in your FID is a small hydrogen flame and dapat na i-present sa inyong collector electrode na kung asa dapat from the name collector, asa i-collect ang katong vaporized na specific particle or molecule. Now, as the sample burns, ions form and then move to your charged collector. So to further explain, thermal conductivity and flame ionization detector. Now, kindly look at this image. This is actually your thermal conductivity detector. Now, asa tong filaments na ginamin diri kanito, 'di ba? Na naa siya filaments na siya ang nag-detect sa changes sa concentration sa inyuhang temperature. And this is how your TCD will work. So, again, na ako'y video na po na i-present. Muna siya mga actual na itsura sa inyuhang TCD, kani and kani. So, again, let us watch this video for you to understand how your TCD works.
How does a TCD work? Each gas enters the gas chromatograph instrument separately. The sample goes into one column while the pure carrier gas goes into another column. Electrically heated resistance wires are located in chambers inside of the TCD. The power supply provides a current to the resistance wires, which causes the wires to heat up. The electrical circuitry shown here that is characteristic of thermal conductivity detectors is known as a Wheatstone bridge. As the gas flows through the TCD, the physical properties of the reference and sample gases, for example, specific heat capacities, will allow the wires of the TCD to be cooled at different rates. This change in temperature will result in a change in resistance from both the reference gas and the sample gas, which produces an electrical signal unique to the compounds being analyzed. This signal is proportional to the concentration of the sample components, providing a direct means of measuring component concentrations in a particular sample. And from this, you receive a chromatogram that represents the components found in the sample analyzed.
Okay, so just to explain further, or at least hatagan ako muog ka ng additional input or more emphasis to how your TCD work, na-mention ka ganina si Wheatstone Bridge. 'Di ba? Now, what is this Wheatstone Bridge? Uh, Wheatstone Bridge is actually, kumbaga murag siya ni ang naga-measure sa ano ng mga resistance values o dira sa inyuhang TCD, and it could also actually be used as for calibration sa mga different instruments o sa mga different na instruments. So that is why it is important to place a Wheatstone bridge, especially in your TCD. So, number one, ang usual na mention po dira sa video na usually the carrier gas that is being used is your helium. Now, if napansin ninyo sa video, nato'y duha ka entry o duha na kung asa pwede mag-enter ang inyuhang sample. The other one is for your standard or reference. The other one is for your sample. Now, as your reference and your sample enters each component or each compartment rather, each compartment, syempre naman tay heat na i-introduce. As mentioned by Henrys, usually ang kanadyo mga sample components nato, nadyo na siya lower na thermal conductivity compared to our reference. And thus, because of the difference sa ilahang thermal conductivity, na siya makreate na dayon na changes sa electrical resistance na ma-detect dira sa inyuhang filament. And that electrical change, that is directly proportional to the concentration now of your analyte. Muna na causing na sa makita ninyo diri na kaning mga peaks, causing na ay peak sa inyuhang chromatogram. Chromatogram ang tawag ani, na ay peaks na makita mo sa inyuhang chromatogram because of those electrical changes that is happening inside. Okay? Sige. I hope nasabtan to siya. Now we proceed with your FID, your flame ionization detector, which is what is presented a while ago sa katong video as to how your gas chromatography works. Na naadjud mo yung literal na flame na makita here. And ang isa sa important yun po na components in yung FID is to have a hydrogen supply. Muna na jud may continuous hydrogen supply diri, and then sa makita ninyo diri na amoy sample inlet, that is where your sample will enter. Then daratong na mention 'di ba kaganina na collector electrode. And then you have your flame igniter, and then ana na naiyahang simple na component. So for you to understand more as to how your flame ionization detector works. And then again, di ba, mas kini-explain maguna ko na it would be better na ma-picture out with ninyo. So, mo nito siya kung ginaingon na expect that there will really be a lot of videos. And I hope you would appreciate the videos that is being placed. Kay, again, kung ako manggod, mas easier manggod yun natin visual na makita na mga movements. So, kindly watch this video.
Gas chromatography (GC) is a common type of chromatography used in analytical chemistry for separating and analyzing compounds that can be vaporized without decomposition. In gas chromatography, there is a mobile phase which is a carrier gas, such as helium, nitrogen and argon. And there is stationary phase, called a column, which located in an oven where the temperature of the gas can be controlled. There is also a detector that measures analytes in a gas stream attached to the GC system. The flame ionization detector is almost universally employed, where the flame commonly is generated with hydrogen and air. The autosampler provides the means to introduce a sample automatically into the inlets. Also, manual insertion of the sample is possible. The column inlet, or injector, is attached to the column head and provides the means to introduce a sample into a continuous flow of carrier gas. In the injector, a sample is introduced into a heated chamber via a syringe through a septum. The heat facilitates volatilization of the sample and sample matrix. The carrier gas then either sweeps the entirety, splitless mode, or a portion, split mode, of the sample into the column. In split mode, a part of the sample carrier gas mixture in the injection chamber is exhausted through the split vent. In splitless mode, all the sample carrier gas mixture in the injection chamber is transported through the column. Two types of columns used in GC: packed column, where the stationary phase is coated directly in the column, and capillary column, where the stationary phase is coated with the inner wall of the column. The mixture separation is based on differential partitioning of the components between the mobile and stationary phases. The component which has less affinity to the stationary phase, consequently less interaction, travels faster and eluted out first. And the component which has more affinity to the stationary phase, consequently more interaction, travels slower and eluted later. In addition, other factors influence the separation of the components, such as column temperature, carrier gas flow rate, column length, and amount of material injected. As compounds elute from the column, they interact with the detector. Different types of detectors can be used, such as the flame ionization detector, thermal conductivity detector, and mass spectrometer detector. The operation of the flame ionization detector is based on the detection of ions formed during combustion of organic compounds in a flame, which generated by hydrogen and air. To detect these ions, two electrodes are used to provide a potential difference. The positive electrode doubles as the nozzle head where the flame is produced. The other, negative electrode, is positioned above the flame. When an organic compound is mixed within the hydrogen flame, mainly carbon ions are generated. And a current is produced between the electrodes proportionally to the amount of organic compound present. This current is measured with an electrometer, amplified into proper voltage, and fed into an integrator. The manner in which the final data is displayed is based on the computer and software. The number of peaks present can indicate how many components are in the mixture. Usually, the x-axis of the gas chromatogram shows the amount of time taken for the analytes to pass through the column and reach the FID detector. Typically, the y-axis, or the area of the peak, is a reflection of the amount of a specific analyte that's present.
Okay, so that's it for your flame ionization detector. So at least medyo ba na-visualize ninyo na there is vaporization that will evaporation that will happen, 'di ba? Na dapat ano sa gyud ninyo ang inyong sample, and then collector electrode will then get those kato na certain evaporation na nahitabo sa inyong sample. And then, syempre, naman dyan ay difference sa inyuhang na kanang na ma-detect sa inyuhang sa atong detector na difference in terms of unsa ba structure, molecular structure, so ing ana. And at least here in the video, it was further explained the difference between your pack column and your capillary column. And katong gipakita gyud niya after explaining is actually an example of your capillary column, since kung makita ninyo, di ba, empty ang sulod, wala sila gibutang na any na beads, coating lang sa boundaries or sa boundaries sa inyuhang column. And syempre, in terms of the movement of your substances, na explain man po sa inyong video na 'di ba, it could either be due to the solubility of your sample. Na syempre, ang inyong man yung mga different components sa isa ka sample na na sila yung difference in terms of solubility. Pero syempre, para makuha nato kung unsa lang gito na ah, specific na component na gusto nato ma-detect, mag-matter na gyud na siya sa kung saan ato ang gamiton or ibutang or i-incorporate sa inyong mobile phase, stationary phase, and sa inyong eluent as mentioned a while ago. Okay, so we are done with gas chromatography. Now we proceed with your liquid chromatography. Now, liquid chromatography uses lower temperatures for separation, achieving better separation of your thermolabile compounds. Now, when we say thermolabile, diba, meaning your compound or sample is actually unstable when heated and they are readily destroyed or deactivated, especially when introduced with heat. Now, in liquid chromatography, easier to recover a sample compared to gas chromatography. And actually, this is a common sense since, di ba, if mapansin nyo, ang ginabuhat biya yun nato kay gas chromatography is i-evaporate yun nato ang ato ang sample. Here in your liquid chromatography, there is no need for that. And actually, liquid chromatography is commonly used and is suitable alternative sa inyuhang suitable alternative kaysa sa inyuhang gas chromatography kay as mentioned also, ako lang po siyang i-emphasize balik na dili man jud tanan compounds are volatile. Some are too unstable and some are insufficiently volatile to be assayed using your gas chromatography. So that is why preferable si liquid chromatography. Now, the mobile phase can be removed and the sample can be processed further or reanalyzed under different conditions. Now, this is actually another advantage of your liquid chromatography over your gas chromatography. Since ma-recover man ang inyong sample, then further processing can be done. Now, there are different types of separation technique using your under your liquid chromatography. So you have your adsorption, partition, steric exclusion, affinity, and your ion exchange. Now, sa mapansin ninyo, here enters your, di ba, adsorption and your partition. O basig nakalimot, di ba, na-mention nato ni siya kaganina dito sa inyuhang gas liquid and sa inyuhang gas solid types of chromatography. Now, that is why wala na nako to give further explain because the principle behind that is actually here na sa pag-discuss nato sa types of separation technique ninyo. Na basig moingon mo, ma'am, daghana ba ana na separation technique? It lima siya ka buok, but actually the principle behind each of the separation technique is very easy and dali lang jud kay siya masabtan. Maka maingon na ninyo later kung ako na gyud siya ma-start og discuss. To start with the adsorption chromatography or absorption type of separation. From the word adsorption, when we say adsorption, what we are trying to look here is the adhesion of your atoms or it could be your molecules to a certain surface. Now, adsorption is an example of your liquid-solid chromatography. Always have ang first na word is referring to your mobile phase, ang second is referring to your stationary phase. So, since liquid-solid chromatography mani, mobile phase is your liquids, stationary phase is your solid. Competition between the sample and the mobile phase for the adsorptive sites on the solid stationary phase, that is what is happening here in your adsorption separation technique. And the stationary phase can be acidic polar, like for example your silica gel, basic polar, like your alumina, or non-polar, like your charcoal. So again, that would depend on the analyte of interest na kung asa siya mas better ma-retain or asa siya better na mag-adhere. Now, the disadvantage lang yun sa inyong adsorption is that strong retention of many compounds by the supports, making them difficult to elute from your column. So, as you can see here in this image, these are examples of your adsorption techniques. Na again, ang inyuha mang yung mga different na uh, components sa inyong analytes, naman na sila gina-carry na different po ng mga structures and interaction, thus affecting its adherence to your stationary phase. So to further explain how adsorption chromatography works, let us watch this video.
Here, one quick biochemistry basics here. Let's talk about adsorption chromatography. Adsorption chromatography is based on the fact that certain solid materials known as the adsorbents have ability to hold molecules on the surface by the phenomenon of adsorption. The adsorption process usually involves attractive forces like van der Waals interaction and hydrogen bonding. One of the common adsorbents used in adsorption chromatography is silica. Silica has SiOH functional group, also known as silanol. The SiOH group interacts with other functional group of the sample molecules. Different sample molecules binds the adsorbent with different affinity. Hence, when the mobile phase is passed, the molecules having less interaction with the adsorbents are released first, while the molecules having more interaction with the adsorbent are released last.
O, di ba? So that is how your adsorption chromatography works. And di ba na-mention man to na ako kaganina na since ang inyuha mang yung mga different substances, especially inside our body, na mag yung mga different na interactions, so that could actually uh, help in terms of how we could recover one substance to the other. Again, adsorption, adherence, affinity, pwede po, medyo synonym sila na anay, certain affinity to your stationary phase. So just like, unsa to inyuhang nakita sa video, your stationary phase, di ba, nag-example is your silica. So kung kinsa man to na ay higher affinity or higher interaction to silica, then siya ang mas madugay og retain. Next, now we go to your partition chromatography. Partition chromatography is an example of a liquid-liquid chromatography wherein separation of substances are according to their solubility. Again, solubility ang ato ang basis sa separation. So in an organic or non-polar solvent and in an aqueous or polar solvent. Now, here in your partition chromatography, since we are talking about liquid to liquid chromatography, somehow naatay ang principle behind this is that like dissolves like. Solubility man ang ato ang basis. And that polar molecules remain in the aqueous solvent. O, kung aqueous solvent man ang ato ang gamiton, then polar molecules will remain. If it is a non-polar molecule, non-polar molecules are extracted now in the organic solvent. Now, the best example for your partition chromatography is actually your paper chromatography. So you can see here. Kaya siya tinatawag po na paper chromatography because what we are using here as a support or tig-facilitate sa separation is a paper. And I guess every one of us have done this experiment. Kaysa ako, ma-remember gyud kaayo na ako na nagbuhat mo gingani, I think even during elementary or high school, na 'di ba, sa isa ka paper, magkuan mo og marker, ug isa ka dot, and then isoak siya with water, and then through capillary action, 'di ba, musaka man gyud na ang inyong paper and then separating different colors. So that is actually the principle behind here in your paper chromatography, as you can see here. But instead of using kato kung gimension na katong marker or katong pentel pen ninyo na mo ibutang na dot, what we use here in this example is an leaf extract being placed here on the leaf extract spot here in your paper. Basig mangutana mo, ma'am, liquid to liquid chromatography man kaya ni? Yes, it is a liquid to liquid chromatography, though we are using a paper, again, that is just the support. Remember, the leaf extract is a liquid, which is being placed here sa ato ang point of kuan sa inyong paper as ang another liquid, this one. Kumbaga, mura ni siya mag-serve as your mobile face na that will help facilitate and carry your analyte, and then until such time na natay makita na ingani na mahitabo. So, isa sa ato ang bantayan diri na term is kani solvent front. Solvent front actually refers to the distance moved by your solvent na from its point, kani starting point, until maabot siya sa end. So, aning end the part, that is the distance, solvent front, that is the distance between or kung unsa kataas ang movement sa inyuhang solvent. And syempre, if i-try ninyo og visualize, naman gyud ni different separation na dayon sa ilahang sa inyuhang mga different na pigments present here, di ba? Different pigments present in a leaf extract. And unsa nga tuwang tan-awon diri na factor na ngano na aman joy pinakauna or pinakalayo na maka-travel na distance here in your paper, again, that is solubility. That the more soluble your component or your component uh, or your substance, the faster the movement. And since fast ang movement, then the greater the distance traveled. Again, let me state it once again. The more soluble the component, the faster the movement, the greater the distance being traveled. And syempre kay, again, lahi-lahi man gyud ni og solubility. Labi na kani sa atong example, lahi-lahi man gyud ni solubility ang atong mga different na pigments, and thus, a separation will occur. Okay? Now, other than propanone or acetone being your mobile phase, kindly take note also, pwede po ethanol, pwede methanol, or pwede chloroform. Again, depende na po na siya sa kung saan analyte na gusto nato i-separate. Next, now we go to your steric exclusion. Steric exclusion is a variation of your liquid-solid chromatography. Other name for your steric exclusion is your size exclusion chromatography. From the name itself, 'di ba? We will already get an idea as to what is this all about. Size exclusion, meaning the separation is based on the size and shape of your substance or your components. Solid phase here is packed with porous material or beads that separate solutes according to size. So that is why, as you can see here, this is your column, kanino packed with beads, and kanina beads mag-matter na po na siya depende sa kung sa po na nga to ang gusto i-separate or makuha na component. So, syempre, sa makita ninyo diri na side, o kanio, porous ang inyuhang beads, meaning sa manufacturer naman ni siya na naay certain pore size na ibutang dira para katulang yun na certain substance lang po nang able to enter. Syempre, those, para sa niyo, sa makita ninyo sa image, those bigger particles, since they cannot enter inside here sa inyong beads, thus remain outsides, and sila ang pinakauna na ma-remove out sa inyuhang column. So to further explain, oh ha, again, nana po tayo another vid. So to further explain pag yun unsa ning size exclusion, unsa ning mga beads para mas ma-understand pa ninyo more.
Size exclusion chromatography is also called Yell filtration chromatography. These two names may make you misunderstand what is actually happening. In the previous videos, we have talked about chromatography as being a process where an adsorbate is adsorbed to the surface of an adsorbent. However, that is not the case in size exclusion chromatography. In size exclusion chromatography, there is no interaction at all between the molecules and the surface of the porous particles. No adsorption, no nothing. Instead, size exclusion chromatography separates molecules based on their size. A size exclusion chromatography column, or SEC column, is packed with porous particles. Small molecules can enter into the pores of the porous particles, while larger molecules cannot. Thus, smaller particles have a longer residence time in the column and larger molecules a shorter residence time. SEC is typically slower than ordinary chromatography due to the absence of surface interaction, so you need a longer column. As an example, SEC can be used for protein desalting. The salt ions are small and can enter into the pores and gets a longer residence time, while the proteins are too large to enter the pores and thus get a shorter residence time.
Okay, so basig mangutana mo na ma'am, unsa to iyahang ginamin na residence time? Residence time, na iyang gina-refer dire is actually the same with the retention time or retention factor that we defined a while ago. So, na-point out po diri sa video, di ba, the difference between your size exclusion chromatography and your adsorption chromatography. Na, syempre, somehow kung isipon ninyo, wala man siya ging ana kay since ang ato amang adsorption is interaction or adherence or affinity, but with size exclusion, walay affinity as emphasized here in the video, walay affinity na nahitabo, walay interaction. It's just that we're just looking at the size na makontrol nato ang iyahang pag-entry or exit dira sa inyuhang column by taking in kung unsa to ang mag-fit sa inyuhang bead. So muna naghatag siya og example like your protein desalting na in order to separate protein from salt sa size pa lang 'di ba? Size pa lang makita na ninyo and what if si protein na ang ato ang gusto i-measure or ma-measure diri or makuha or ma-separate. Then syempre ang inyong beads dapat as small as kaning sa inyuhang salt kay para tanan salt components enter dito sa inyuhang bead, ma-capture sa inyuhang bead, and then the rest katong mga protein nyo since large man og size, sila lang pud ang ato ang ma-exit out dito sa inyuhang column. Okay? So, di ba? Dali lang kayo ang inyuhang separation techniques. Next is your ion exchange. Now, ion exchange chromatography uses a resin as your stationary solid phase for covalent attachment of anions or cations onto it. So from the name itself, ion exchange, now we are trying to look at electrical charges. Solute ions of the opposite charge in the mobile liquid phase are attracted to the resin by electrostatic forces, and ion exchange chromatography is widely used for the separation of proteins, peptides, and nucleic acids. Because going back, biochem, diba? Going back to biochemistry, proteins na charges. Peptides are made up of proteins. Nucleic acid needs also proteins, or it's made up of proteins. So that is why ion exchange chromatography is widely used in relation to proteins because of the electric charges that it carries. Now, for an ion exchange, a resin type can either be a cation exchanger or an anion exchanger. So dara na siya. Asa ni ako ang ginapsulti na resin type, this one. Cation exchangers have a negative, have a negative charge na resin which will exchange positively charged ions, whereas your anion exchanger na positively charged na resin which will exchange negatively charged ions. And thus, as simple as this one, ang ato ang ion exchange chromatography. Again, we are trying to look at the charges of your components or sample or analyte. Siyempre, 'di ba, opposite attracts. If you place if you place your anion exchanger here, kaniyo, anion exchanger having a positively charged type na resin, kinsa lang man ang ma-attract sa iya ha, then that would be your negatively charged. And thus, all of your positively charged dayon ng mga components will be separated, siya ang una nato na makuha, and then magpabili na dayon ang inyuhang negatively charged na naka-adhere dito sa inyuhang stationary phase or sa inyuhang resin. O, diba? Now we go to the last part, your affinity chromatography. Between the 5 different types of separation technique, affinity chromatography is actually the most selective type of chromatography. As to the reason why, it is because it utilizes specific interactions between one kind of solute molecule and a second molecule that is immobilized on a stationary phase. Some examples of your affinity chromatography are the following: your antigen and antibody, your enzyme and substrate, your receptor and ligand, your protein and nucleic acid. So sa inyo ang mapansin between the pairing, 'di ba? Mura siya katong lock and key mechanism ninyo na only an enzyme can bind to a specific substrate, 'di ba? Only a certain antigen or antibody na ay specific yun na pairing. So that is why it is the most selective type kay because kanina mga substances nato naabi adjuning specificity somehow na property, then makakuha gyud pud ta ani or maka-obtain gyud ta ani mga specific pud kaayo na mga components. So, mo ni actually inyo makita diri na image. 'Di ba? Kung tan-awo nato, mura lang gud pud na siya katong sa inyohang enzymes na for example, enzyme and substrate. So nagbutang ta dahan og mga substrate diri sa inyohang stationary phase. And kung kinsa lang pud to ang kung kinsa to the specific enzyme ang able na makabind ana kay substrate, which happens here, siya lang po na mag-bind, and then the rest kay ato na siyang amo na siyang pinakauna nato na ma-retain. And then at a later part na dayon, pwede na nato ma-measure tong nagpabilin didto na nakabind sa inyuhang substrate. So to further explain, na ako'y video also for this, so kindly watch.
Hey everyone, quick biochemistry basics here. Let's talk about affinity chromatography. Affinity chromatography is a chromatography technique in which separation occurs based on the specific interaction between the two molecules. This specific interaction can be between enzyme and a substrate, receptor and a ligand, protein and the nucleic acid, etc. Let's see different components of the affinity chromatography. The affinity chromatography is usually carried out in column, which is filled with the supporting material or the matrix. The matrix should have the following properties. It should be chemically inert, should exhibit good flow properties, should have sufficient functional groups for the covalent binding of the ligand. Different materials like agarose, polyacrylamide, polystyrene, cellulose, silica, etc. can be used as a matrix. The matrix is attached with a spacer M, which is usually made up of CH2 group. The presence of spacer prevents the non-specific interaction of the ligand with the matrix itself. Let's see the working of affinity chromatography. When the mixture of different molecules is passed through the affinity column, only the molecule that have specific affinity for the ligand binds the ligand. The other molecules are washed away. Next, to disassociate the molecule of interest from the ligand, a buffer with different ionic strength or pH is passed. This finally releases the molecule of interest from the ligand and the molecule is obtained in the pure form. The molecule of interest can also be detached from the ligand by using a competitive inhibitor. The competitive inhibitor can further be removed by dialysis or by changing the pH or the ionic strength. Let's see different examples of ligand and the molecule of interest that can be purified with affinity chromatography. AMP can be used for the purification of NAD-dependent dehydrogenase and some kinases. ADP can be used for the purification of NADP-dependent dehydrogenases. Calmodulin for the purification of calmodulin binding enzymes. Avidin can be used for the purification of biotin containing enzymes. Metals such as nickel can be used for the purification of proteins that have a histidine tag. Lysine can be used for the purification of rRNA.
Okay, so that is what is your affinity chromatography is all about. So again, take note at mga na-mention sa videos. It is also important with regards to the matrix, to the ligand, and how your component of interest is actually removed. So at least na-explain po diri sa video unsaon to siya. Diba? You could either add a certain solvent that could actually change the pH or whatsoever, and it could also be another na kanang substance na competitive inhibitor ato sa inyuhang component, like for example sa inyuhang enzyme ba or sa inyuhang substrate, in order na ma-remove lang gyud tong substance na gusto po nato i-measure. So now we proceed to your chromatographic procedures. So some additional procedures in relation to your chromatography other than katong inyuhang gas and solid. So natay thin layer chromatography and your high performance liquid chromatography. As of the moment, or as of this modern times, HPLC na gid kayo na ang the best. Na even if you do your research, always gina-recommend is for you to do or perform HPLC. You will know later why. Basta ang pinaka first na i-point out nako siya gyud ni ang highest sensitivity na gyud compared to the other pa na mga procedures that is already present currently. Di ba? Kay naa na biya mo research. So, pwede ninyo na siya i-consider. But another factor po na pwede ninyo isipon is gasto siya. Kay actually mahal ang HPLC. So, anyway, going back, first is your thin layer chromatography. Now, thin layer chromatography is a variant of your column chromatography. Kaya siya ginatawag na thin layer because we are placing here a thin layer of either a sorbent, a thin layer of sorbent of either could be an alumina, silica gel, cellulose, or cross-linked dextran, which is uniformly coated now on your glass or plastic plate. So, thin layer chromatography is most commonly used as a semi-quantitative screening test. And this is how your thin layer chromatography looks like. Now, ma'am, mura man lagi siya og paper chromatography. Actually, almost the same gid ni siya with paper chromatography, but instead of using a paper, yun na yung thin layer na sorbent. Na ang ibutang na nato dira is katong inyuhang silica gel and all. And then same principle will now apply. Na kung makita ninyo, kanina siya ang formula sa inyong retention factor. So matingala mo unsa ni. And then kani, naalang pud mo'y mo ni siya inyuhang mobile phase. So, diri ninyo i-place daan ang inyuhang analyte. And then, syempre, after some time, through capillary action, na separation dayon na mahitabuan sa ilaha. Sa inyuhang components rather. Nga na lang na po ang inyuhang thin layer chromatography. How about your high performance liquid chromatography? Now, high performance because natay mga gipang-add diri na components that could hasten kaniya uses pressure for fast separation. So meaning na atay gi-add diri na makapapaspas pa further na ma-separate ang inyuhang components, na atay controlled na temperature, na atay mga inline detectors, and then atay gradient elution techniques na gi-add pa sa iyaha. So that is why it is called as such. So ingani ang inyuhang component or setup sa inyuhang HPLC. And to further discuss how HPLC works, kay ako alam man po ding isa-isa ako nag-discuss sa inyuhang basic components. So kindly watch this video first.
Here, one quick biochemistry basics here. Let's talk about HPLC. HPLC is also known as high performance liquid chromatography or high pressure liquid chromatography. HPLC is usually a modified column chromatography. In column chromatography, a column is packed with an absorbent material like silica, and the mobile phase is passed down the column because of gravity. But in HPLC, a high pressure pump is attached with the column. The high pressure pump can generate a high pressure up to 40 megapascals. The column is filled with an absorbent material which has a very small particle size. The small particle size gives a very large surface area for the sample molecules to interact. As a result, the efficiency of separation increases, giving high resolution. Let's see the components of HPLC. The column is made up of stainless steel which can withstand a very high pressure up to 50 megapascals. The length of the column can vary from 5 to 25 centimeters and have an internal diameter of 4.5 millimeter. The flow rate of mobile phase through the column is usually 1 to 3 ml per minute. The stationary phase. As discussed earlier, the stationary phase is made up of an adsorbent material that has a very small particle size. The particle size is kept uniform to obtain a better performance. Usually chemically modified silica, divinyl benzene, etc. are commonly used as a stationary phase. The mobile phase. The mixture of different solvents can be used as a mobile phase. The solvent used depends on the type of sample molecules which are to be separated. The mobile phase solvent can be polar or non-polar. The mobile phase is usually kept in the solvent reservoir. The solvent reservoir is attached with a pump which pumps the mobile phase in the column with a high pressure. Just before HPLC column there is an injector which allows introduction of the sample in the column. The detector. In order to detect the eluate, that is sample coming out of the column, the HPLC column is attached with a detector. Different types of detectors such as UV detector, IR detector, fluorescence detector, refractive index detector, mass spectrometer, electrochemical detectors, etc. can be used. The detectors are connected with a computer which collects the information. Now let's see the working of HPLC. As the sample molecules get separated, they are detected by the detector and a peak is obtained on the computer. This peak is plotted with respect to the retention time. To identify the components, we need to have standards. Let's understand this with an example. Let's say we run glucose as our sample and the peak of glucose is obtained at 5 minutes. Next, we run sucrose as our sample and the peak of sucrose is obtained at 8 minutes. Now we analyze the unknown sample for the detection of sugars present in it. Many peaks are obtained in the chromatogram. The peak obtained at 5 minutes indicates the presence of glucose. However, there is no peak at 8 minutes which indicates that sucrose was absent in our sample. The remaining two peaks are still unknown as we don't have any standards to compare their retention time. This is how the detection of sample is done in HPLC, and for the detection we must have the standards.
Okay, so additional na learning here in this video is actually in terms of the purpose of your standard and identifying your sample. If nagalab gyud unta mo karon sa inyong chem, you would appreciate more the purpose of your standard. Since every time you try to measure a certain a certain na kanang substance, like for example, kani glucose, your lipid, always gud mo na magbuhat og standard. From the name itself, standard meaning siya ang ato ang basis na para makabaluta na ang kanig yun inyuhang sample or substance na gusto i-measure your analyte is mo gyud na siya ang present. And naalam po ani na certain na concentration. Na as to the laboratory, since you are talking about normal ranges, muna na magranta og standard kay para kabaluta na dapat nalang yun diri within the range. And that is how your HPLC works, diba? So, to further discuss the different components in your HPLC, na-mention kaganina na atay high pressure pump na ginagamit. So, what is this pump? Pump forces the mobile phase through the column at a much greater velocity, and these are examples of the different pumps being used. Now, again, kaya siya high pressure pump helps in hastening separation. And there are three kinds. Your mechanical reciprocating pump, which is the most widely used. Pneumatic pumps for pre-operative purposes. You have hydraulic amplifier pumps, but as of the moment, it is no longer commonly used. Next, we have the column. So we already know the purpose of the column. And here in your HPLC, it is a long stainless steel. And the most commonly incorporated in your column is your silica gel. Next, we have your sample injectors. Diba nakita dito sa video na other than the high pressure pump being directed, natay isa pa na i-insert dito sa inyong column, which is your sample injector, where you will insert or place or introduce your sample. So it can be used to introduce the sample into the path of the mobile phase that carries it into the column. And actually, natay isa ka example sa inyong sample injector, and that is your loop injector, which is the best and most widely used na type of sample injector. Why? Because it has high reproducibility and it can be used at high pressures. Now, na ako'y video diri na ipakita sa kung unsa ning itsura sa inyong loop injector. Expect na wala ni siya sound. So just try to observe and read kung sa man ang mga captions na ibutang, then try to appreciate also how your loop injector works. So you see, that is how your loop injector works na depende sa kung unsa ang ato ang gusto or padulungan sa ato ang sample. So switch lang siya. Is it going to the column or going to the vent or waste na ba siya? So kana lang turning off your sample injector depending on where your sample should go. Next is your detectors. Now, syempre, always kailangan yun ni si detector. It monitor the eluate as it leaves the column. It produces an electronic signal proportional to the concentration of each separated component. Now, as to the detectors, it can be either of the three. Pwede spectro, which can detect absorbances of visible or UV light. Pwede photodiode array, which is used for spectral comparisons and compound identification and purity, and for drug analysis. And lastly, your amperometric or electrochemical detector, which measures current produced when the analyte of interest is oxidized or reduced at some fixed potential set between a pair of electrodes. So, syempre, depende lang yun na siya sa kung unsa ang ato ang analyte pod na i-measure, na kung mogamit pa tag spectro, diode, or kaning amperometric or electrochemical detector. Lastly, I think this is the last. Last is your recorders, which is used to record detector signal versus the time mobile phase passed through the instrument starting from the time of sample injection. And kanina mention na po na ako ni sa inyo ha, katong graph na inyong makita is what we call your chromatogram, and that the peak area is proportional to the concentration of the compounds that produced the peaks. So, we are done with your chromatography. Now we go to the last part of your analytical techniques. There is ako na discussion, your mass spectrometry. Now, there may be cases na ang inyuhang makita na term is mass spectroscopy. Actually, they mean the same. Kanang inyuhang liquid or gas chromatography, actually always na siya ginakouple with your mass spectrometry. Muna na naamoy madungga na GCMS, LCMS, mass spectrometry yun na ang combined. And usually sa mga, labi na sa mga kaning current na to or sa modern times karon, it is actually considered as the gold standard for most testing jud kay ang pag-couple manggod sa inyuhang gas chromatography with this mass spectrometry kay it actually doubles or mas ginapataas pa niya ang sensitivity and specificity. So, syempre, kung mutaas ang sensitivity and specificity, then the better. Okay, that would also mean na more accurate and precise ang atuang testing. So, unsa day ni mass spectrometry? Now, mass spectrometry is based on fragmentation and ionization of molecules using a suitable source of energy. Charged particles moving through a magnetic or an electrical field can be separated from other charged particles according to their mass to charge or MZ ratios. So to emphasize that your mass spectrometry measures or separate your different components according to the mass to charge ratio. So what are the basic components now of your mass spectrometry? We have your sample inlet. Of course, dara na to i-inject ang ato or asa na to i-sulod ang inyuhang sample. We have your ionization source, your mass analyzer, and then your ion detector. Now, kaning electron ionization, this is actually a method that requires a source of electrons in the form of a filament to which an electric potential is being applied. So the actual measuring of your MZ ratio, your mass to charge ratio, occurs when the gas phase ions pass into your mass analyzer. And then, as it passes through your mass analyzer, it will generate an electrical field that can manipulate the charged molecules to sort them now according to their mass to charge ratios. And the most common means of detecting your ion actually employs an electron multiplier. So this is the how your mass spectrometry would look like. But actually, pinaka-simple na ni siya of way of presenting the components of your mass spectrometry. Na kanina amoy sample inlet, turain yung ionization source, then your mass analyzer, and then your detector. And then, of course, nakagapon tong recorder. Now, mass spectrometry actually involves four major steps. One is the conversion of the parent molecule into a stream of ions, usually singly charged positive ions. The next is the acceleration of your ions into a magnetic or electrical field. Then separation of your ions by mass to charge ratio. And lastly, counting of the number of ions of each type or measurement of current produced when the ions strike a transducer or labi na kato sa inyuhang detector. So there are actually two types of mass spectrometry, starting with your quadruple mass spectrometer. Now, direct electrical current and radio frequency voltages of selected magnitude are applied to two pairs of metallic rods, and only ions of specific mass to charge ratio can pass undeflected to the end of the rods where they are detected. And this is how your quadruple mass spectrometer would look like. Kani na siya actually na part. Kaya siya quadruple, kay upat na siya ka rods na gidikit-dikit and naka-pair na siya na duha ka rod positively charged, duha ka rod negatively charged. So to better understand how your quadruple mass spectrometer and how its component will work, so tanaw na po ta og video ha.
What happens if you have something like this, and you want to find out if something like this is inside? Obviously you can't tell just by looking at it, but if you separate things out, the answer becomes clear. Scientists have the same problem. How do you know if there was once water on Mars? Or, for that matter, life? Obviously, you can't tell just by looking at pictures of Mars, but scientists think the answer may lie hidden in tiny molecules in Martian soil. So, how do you take apart a molecule to see what's inside? Luckily, scientists have a tool to do just that. It's called a mass spectrometer, and it lets us take an extremely close look at whatever we're studying. And even though Mars immediately comes to mind, mass spectrometers are used in multiple NASA missions. They're also used in labs for hundreds of scientific purposes. But the important question is, how does it work? Today we'll be looking at a special kind of spectrometer called the quadrupole mass spectrometer. It's called this because of the 4 long poles that make up the center of the instrument. So say you have a sample that's been turned into a gas, and you want to find out if it contains certain things. The gas is sent into the mass spectrometer, first hitting a piece called the ion source. Here, a stream of electrons hits the molecule, breaking it into fragments and giving each fragment a charge. Next, the fragments enter what's called the analyzer. Here, they're separated based on their mass, and the analyzer is tuned so that only the fragments we want to see make it all the way through. Everything else flies off in a different direction. After this, the fragments hit what's called the detector, and scientists record the data. If you're looking for more than one kind, the analyzer can scan across a range of fragments, building up a record of not only what kind, but how many. Once you have these results, called a mass spectrum, you can verify that your sample in fact contains what you're looking for. And here, the real work begins. The mass spectrometer is a powerful tool, and by taking many samples, looking at the results, and studying what we find, scientists can work to discover not only the secrets of water and life on Mars, but also answers to bigger questions about the universe. And all by studying something as tiny as a molecule.
So that is how your quadruple mass spectrometer works. Again, as depicted in the vid, separation is based on the mass to charge ratio. If it's not the analyte that you are looking for, then it is being deflected in 2 different directions, and the only thing that could pass through is your analyte of interest. So, di ba, grabe gyud ka technical ani sa ato ang instrumentation. So, muna na I would prefer to present certain videos for you to really picture out everything. Second type is your ion trap mass spectrometer, wherein there are 3 electrodes present in a ring shape and 2 end caps, produces ions in the cavity until selectively ejected to the ion detector. And this ion trap mass spectrometer has the ability to get full mass spectra at very low sample concentrations. Now, actually wala ko'y makita na vid for this sa inyong ion trap mass spectrometer, but murag ang ginamit lamang yun ani is from the name itself ion trapping, but as to how it exactly works, di kay na ko siya ma-explain yun. So what I would like to present na lang for you is regarding its component. So mo ni siya, this is your ring electrode. Kaya siya 3 electrode because kaning duha ka end cap here is still an electrode. Murag end electrode actually ang tawag ana niya. And again, murag ang basic principle lamang gyud ani sa inyong ion trap is trapping of your ions here by using your 3 electrodes. But then again, that's all you need to know. And other than that pa di ay, ang isa sa ginatanaw nato diri sa inyong ion trap mass spectrometer, other than the master charge ratio, is that there is a direct current and a radio frequency that is needed. But, other than that, kato lang jud ang ako ang ma-emphasize na sa inyo ha. Now, another type of mass spectrometry is your tandem mass spectrometry, wherein, so kanina siya, I think you are all familiar with this, since kanina siya ang common na ginagamit during drug testing, na kaning GCMSMS, LCMSMS. So, saan ginamin ani, meaning tandem kay tinigduha ka mass spectrometry ang present, and this tandem mass spectrometry is used for greater selectivity and lower detection limits. Na kumbaga, even a very low na jud na level, masin siguro 1 lang na kabuok na analyte, kaya gyud kay niya i-detect. So, here in your tandem mass spectrometry, we link 3 quadruples here in a series, now called as your triple quad. The first quadruple is used to scan across a preset of MZ range and select an ion of interest. The second quadruple functions as a collision cell, and the third quadruple serves to analyze the product ions generated in your quadruple 2. So meaningful product ion scan or selected reaction monitoring is being done. And this is how your tandem mass spectrometry looks like. So, mao na dahil ni siya na ionization na mahitabo, mass analysis, dissociation, and then another mass analysis, then detection. And then this is how your triple quad will look like. Oh, so mo ni siya. So if we will arrange that one, so ingani ang iyahang whole na itsura na makita. So that actually ends our discussion, kids. So thank you for listening all throughout. We'll be posting the individual links of the vid just in case na gusto ninyo siya tanawon separately.